Welcome, everyone, to this evening's lecture. Down in London, should you ever go there, if you're history and philosophy of science minded, you head down to the museums area at South Kensington, and eventually you hit the main drag. And then you have to make a choice. Either you go left to the Natural History Museum, or you go right to the Science Museum. That's one way for organisms, another way for objects. Armadillos over there, air pumps over the other way. Now, those of you who've been attending these lectures or following along online will have noticed that at Leeds we don't do it like that. We haven't made that choice. Our collection integrates organisms and objects together. Now, that's partly an accident of how we happen to inherit what we've got. But it's better than that because it fits with our conception of our field, in which the history of the life sciences and the history of the human sciences are at least as important as the history of the physical sciences. And we've tried to design these lectures accordingly. Now, that said, over the last run of lectures, objects have been in the focus. We hope they've been interesting objects. We've had the first economics computer, the Newland Phillips machine, the X-ray camera, which took the first photograph of DNA. We had a stethoscope, which very possibly made by the inventor of the stethoscope, Linneck. And last month, we had the nearest thing that anyone's ever made to a working model of the Leeds perpetual motion machine, which used to be hawked around county fairs, these parts in the 19th century. But tonight, for the 14th lecture in this series, pardon me, organisms come back into the spotlight. Our organisms, which are also, of course, objects, are corals. Our lecturers are Dr. Ellen Clarke, a specialist in the philosophy of biology, and Emily Herring, a PhD student who's a specialist in the history of biology. And as their lecture title promises, we'll be getting lessons from the shallows, from this fragile enigma. So please join me now in welcoming Ellen and Emily.
Thanks very much, Greg, and thanks to everybody for coming out on this rather grim evening. Emily and I are going to transport you to a tropical paradise to hopefully cheer you up a bit. So, the word coral is used in two ways. It's used to refer to this intricately patterned, stony stuff, and it's also used to refer to a class of organism. The organism is a small marine invertebrate. It's one that's closely related to sea anemones and jellyfish. All of the organisms in this group, which are called Cnidaria, spend their early life floating around in the ocean with their tentacles down like this in what's called a medusa stage. And then some of them, including coral, settle on a rock upside down. So a coral polyp is essentially a very small jellyfish. They're usually around a few centimetres long. It's a very small jellyfish doing a handstand on a rock with its tentacles waving in the air. So this is what it looks like when the coral polyps extend their tentacles. It looks like a sort of garden of small wriggling flowers. Ooh, I need to press play. No, you can see that? The colour's a bit odd, never mind. There are 2,500 different species of coral, around half of which are stony or reef building. So as they grow, these stony corals lay down layers of calcium carbonate underneath and around themselves. They grow, they put a layer of calcium carbonate down, and it sort of forms a protective cup or exoskeleton into which the polyps retreat by day and extends their tentacles at night to catch food. The calcium carbonate protects them, and it also very slowly sort of jacks them up into the water column and higher up into the light. When they do this in huge numbers, they build coral reefs. So these are the sort of undersea cathedrals of solid calcium carbonate, which can be as big as mountains. There's a huge variety of different coral architectures. Some of them look like brains or like the branches of a tree. Some of them look like dead man's fingers, or like the petals of a beautiful underwater rose. Some of them are encrusting or plate-like, shaped like tables or boulders or delicate fans. And the different forms are linked to different habitats. So they depend on things like the the availability of light, and how much the water moves in that location. Now, as well as being really beautiful and making for fantastic scuba diving photos, corals are very, very cool. We often regard animals in this group as sort of primitive or simple, just a sort of ring of tentacles surrounding a stomach. But this is wrong. They have what Richard Dawkins calls probably the most complicated piece of apparatus inside any cell, anywhere in the plant or animal kingdoms. So this is a nematocyst. And essentially this is a cell containing a tiny spring-loaded harpoon attached to a thread. So it sits coiled up inside the cell under osmotic pressure, and it's got a hair trigger, so when something touches the hair trigger, the cell bursts open, shooting the harpoon out and into its victim. This is what makes corals and sea anemones sticky to the touch. If you've ever touched one, they feel sticky. It's all the little harpoons sticking into your fingers. Although some of them inject potent poison, so don't do that too much. The corals use their tiny harpoons to capture food, floating plankton, and drag it into their central mouth, as well as to defend themselves from predators. They can also turn their stomachs inside out and extrude digestive juices to fend off encroaching neighbours. And they also know how to have a good time. Most corals engage in something called broadcast spawning, so this is the synchronized release of huge numbers of eggs and sperm into the water. And because they do it at night, usually around the time of a full moon, it creates this really beautiful undersea snowstorm or starscape. Often causes a feeding frenzy as well for fish and other filter feeders like whale sharks. And it's done in such great numbers that it produces great colourful slicks of colour on the ocean like this as well. So, they're cool animals. What Emily and I want to do today is impart to you some sense of the extent to which corals are enigmatic creatures, creatures that really confound our tendency to want to place life into neat little boxes. Emily's going to tell you a historical story in which corals have confused us from ancient times right up to the birth of the modern synthesis. And then I'll pick up and tell you about two contemporary conceptual puzzles in which corals have a starring role. Corals have inspired a range of emotion down the ages, including fear and confusion, as well as awe and affection. And there will be some sadness too, as I tell the story of the trouble that corals are now in. But for now, over to Emily.
Thank you, Ellen. Can everyone hear me properly? So, now that we're all on the same page about what coral is, I'm going to take you through an accelerated tour of several centuries of reflections about the identity or identities of corals. So if you look at these three categories, animal, plant, or mineral, usually we don't really struggle, given any object or organism, to ascribe them one of these categories. But for some reason, with coral, it hasn't always been that easy. For a very long time, naturalists thought that coral was either a mineral or a plant. Aristotle did have a category for animals that resembled plants, or animals or organisms that were in between plant and animal life, and he called these zoophytes. I don't think this is exactly what he had in mind, but it's probable that he put coral in this category. However, until the 18th century, as I was saying, most naturalists thought of coral as either a mineral or a plant. Roman poet Ovid added his own mythological take on the origin of coral in the 4th book of his Metamorphoses. So Perseus is the hero who'd succeeded in the seemingly impossible task of killing Medusa the Gorgon. So she's the snake-head monster who turns anyone who looks into her eyes into stone. And there he is holding her head. And during his journey home with the Gorgon's decapitated head in his bag, he had to confront another terrifying monster, a sea monster, which he killed using the head, turning it into stone. And Ovid says that after this victory, Perseus washes his hands in the seawater, and so that Medusa's head, covered with its snakes, is not bruised by the harsh sand, he makes the ground soft with leaves, and spreads out plants from below the waves and places the head on them. The fresh plants, still living inside and absorbent, respond to the influence of the Gorgon's head, and harden at its touch, acquiring a new rigidity in branches and fronds. And the ocean nymphs try out this wonder on more plants, and are delighted that the same thing happens at its touch, and repeat it by scattering the seeds from the plants through the waves. Even now, corals have the same nature, hardening at the touch of air, and what was alive under the water, above water, is turned to stone. And so this is the scene that you can see here depicted in Giorgio Vasari's painting. And I just think it's interesting that Ovid had to resort to a myth to explain coral's ambiguous nature, which he assumed to be something between plant and mineral. And the story seems to have given its name to an organism that's very closely related to coral. So this is a Gorgonian colony, which does look a lot like the petrified, if you can see the ocean nymphs, what they're holding, it does look like the petrified sea plants in the depiction of the myth. I don't know that this is actually where the organism got its name from, but I choose to believe that it is. I like to believe that it is. And looking at these pictures, you can sort of see why Ovid and others would think that corals were something between minerals and plants. The branching structure of certain corals and their flower-like structures, like this specimen on the left, made people think that it was a plant. And if you only had access to a dry specimen, which is essentially what is left of the exoskeleton, that's sometimes colored by organic pigments, like the specimen on the right, you will have the impression that it's some sort of beautiful mineral formation. But if you were early 11th century Iranian polymath al-Biruni, you might think that they are in fact animals after all. Al-Biruni hypothesized that organisms such as sponges and corals were neither plant nor mineral, but were in fact animals because they responded to touch. But this idea didn't catch on until the 18th century, with the experiments of a French physician called Jean-André Peyssonnel, who was born in Marseille in 1694, and he studied medicine at Aix-en-Provence. And he became known for his work on the plague and ways to avoid contagion, but he had a hobby on the side doing that. He lived on the Mediterranean coast of France, which allowed him to go and study the marine wildlife, including corals, and he did what no one had done before. He extracted corals, and he tested them in a variety of conditions. He boiled them, he dried them, he poured acid over them, and he noticed that they expanded and retracted, not only when they were extracted from the water, but that they also reacted to touch. And so he interpreted this as a form of irritability, which was considered by some at the time to be one of the marks of animality. It took a while for his theory to be accepted, but he eventually convinced the Académie des Sciences in Paris that corals were, in fact, animals. Even the immensely famous naturalist Buffon, in his 1749 Histoire naturelle, agreed. What we first took for minerals were then classified as plants, and now and forever belong to the animal kingdom. So the notion that corals were animals and not plants was therefore established in the first half of the 18th century. And a bit later in the century, in the 1770s, it was proposed that the huge coral reefs that the navigators had observed during their travels were the result of the growth of the animals. So coral reefs have in the recent decades become symbols of the devastating effects of human-caused global warming and pollution. However, before the 20th century, coral reefs were seen as objects of wonder and fear. In the 18th and 19th centuries, they were seen as dangerous for those navigating the oceans. So we currently distinguish three types of coral reefs. The first type is a fringing reef. So they grow around islands and continents near the coastline, and they're separated from the shore by shallow lagoons. The second type is a barrier reef. They also parallel the coastline, and they're separated by deeper lagoons. And at their shallowest point, they can reach the water's surface, forming a barrier to navigation. The Great Barrier Reef, is that me? Sorry. Which covers 2,300 kilometers off the coast of Australia, is a system of almost 3,000 individual reefs. And it's in fact the largest organic formation, and you can see it from space, so it's pretty big. And finally, atolls, which are rings of coral that encircle lagoons and are usually located in the middle of the sea. And so Ellen explained in the introduction how the coral polyps gradually build coral reefs. And the growth rate of coral varies depending on several factors, water temperature, food, but depending on the size, it can take between 100,000 and 300,000 years for a barrier reef or an atoll to form. And since we're talking interesting facts, here's another interesting fact about a slow and gradual process related to coral. So 70% of the beautiful white sand that you can find on beaches near coral reefs is in fact composed of tiny bits of coral. So what happens is there's a fish called the parrotfish that swims around and eats polyps and all sorts of things. And it also eats hard coral, and it breaks it up into tiny bits with its teeth, but it can't really digest it, so it excretes it immediately. So what I'm essentially saying is, next time you're lying on a beach, a beautiful tropical beach, just bear in mind you're actually lying on a pile of parrotfish droppings, that one specifically. But back to the history of the study of coral. In the late 18th and early 19th century, coral reefs were gaining a lot of attention, firstly because they constituted a real hazard. If visibility was low or the sea a bit agitated, they could become invisible to ships and therefore very dangerous. And in addition, the growth rate of coral reefs was not known at the time, so some naturalists started panicking and speculating that these huge formations constructed by coral polyps could become new continents rapidly emerging and potentially blocking the routes of maritime commerce or changing the submarine topography, which would make it difficult to navigate. And so the naval surveyors of the time developed an obsession with coral and coral reefs. And when plans were made for the second voyage of the HMS Beagle in 1831 under the command of Captain Fitzroy, among the surveys that were meant to be carried out was the study of coral reefs. This had become a standard order for the Hydrographic Office of the British Admiralty. And among the orders that Captain Fitzroy received, he was told to exert, I quote, every means that ingenuity can devise to find out how the circularly formed coral islands in the Pacific came to be. And if you've been paying attention at all, you will have guessed that the circularly formed coral islands referred to atolls. And the young Charles Darwin, who'd been invited to be a naturalist on board the ship, was asked to reflect about how these circular reefs were formed. And you have to admit, they are kind of weird and mysterious and almost eerie, like these rings of coral in the middle of the sea. It was very mysterious. Where did they come from? How deep under the water did they go? People just were very confused by them. And so this was the mystery that Darwin had to solve. And during the journey, Darwin has been reading the first volume of Charles Lyell's Principles of Geology. Lyell described geological change as steady and continuous accumulation of tiny changes over time. And the appearance of the Earth's crust could be explained by slow movements of subsidence and elevation. And during the early stages of the Beagle voyage, Darwin made geological observations that he thought confirmed Lyell's theory. The main theory for the formation of atolls, to which Lyell subscribed, was that they grew on top of submarine volcano craters that were lifted from below towards the surface. In November 1835, Darwin observed this island. It's currently called Maria, and it's part of French Polynesia. And he observed it from high up on a neighboring island. And that's when the idea struck him that perhaps instead of being elevated from below, so the atolls were actually the result of something sinking. So the idea was, if a landmass which has a fringing reef growing on its shore sank at a slow rate due to continuous geological forces at work, the corals would be able to maintain growth so long as their growth rate was faster than the rate of the sinking. And if the landmass that was sinking was a circular island, then once the mountainous structures upon it had completely disappeared under the sea, all that would be left would be this circular formation. In other words, an atoll. And so Darwin published his results in 1842 under the title The Structure and Distribution of Coral Reefs. And this constituted one of his lesser-known triumphs, because this is more or less the theory we still have today. And when Darwin started putting together his theory of evolution, writing his notes for himself in view of the origin of species, his research on coral was clearly still in his mind. He famously wrote in one of his notebooks that the tree of life should perhaps instead have been called the coral of life. And so the idea was that the branching formation of coral better represented the history of life, because while the trunk and roots of a tree are still alive as the branches grow, with coral, or at least with the branching species of coral, each living polyp is continuously and gradually building upon the dead skeleton below, just in the way that current species are building upon thousands and thousands of extinct species. But whether we choose to represent the evolution of life with a branching tree or coral, and as you know, Darwin finally stuck with the tree model, the diagram works in the same way. So the points joining the two divergent branches represent common ancestry between two separate phyla. And this idea of common ancestry got a French philosopher called Henri Bergson to question the real difference between animals and plants. The existence of plant-like animals like coral blurred the lines between animality and vegetality, which is a word, and this apparent overlap between the two kingdoms took on a whole new meaning once inserted within an evolutionary framework where taxonomic groups were perceived as historical entities rather than static creations. So in 1907, Bergson published his own metaphysical take on organic evolution, L'évolution créatrice, creative evolution. And in the second chapter, he asked the following question: what characteristic best defines animal life and distinguishes it from plant life? And he reviewed the various characteristics that were usually used to distinguish between the two. So was it different modes of alimentation? So the difference would be that whereas plants make their own food, synthesizing it through photosynthesis, animals have to seek their food outside themselves. So in modern terms, we'd say that plants are autotrophic and animals are heterotrophic. However, there exists a whole range of plants that do not fabricate their own food, or don't always need to, like parasitic plants or carnivorous plants. So was it movement? We tend to think of animals as mobile and plants as very much sedentary. But once again, said Bergson, movement was not a good criterion to distinguish between animals and plants, because in that case, a whole lot of animals would be classified as plants. So think oysters or corals, or even this very beautiful sponge that's a very weird color of blue. And then following this argument, Pissonell's criterion of irritability to characterize animals wouldn't work either, because there are plants that actually react to touch. And that pink flower you can see right there is one of them. Mimosa pudica actually reacts to touch. So according to Bergson, even consciousness was not a good criterion to distinguish between plants and animals. And he concluded, no definite characteristic distinguishes the plant from the animal. There is not a single property of vegetable life that is not found in some degree in certain animals, not a single characteristic feature of the animal that has not been seen in certain species or at certain moments in the vegetable world. And as I said earlier, these reflections tied into his reflections about common ancestry. He was thinking about the branching nature of evolution, and the fact that no matter how different two phyla may be, you can always trace them back to their shared common ancestor. He believed that if we looked back far enough, we would find the common ancestor for all plant and animal life, and that even though we didn't know exactly what it would look like, we could imagine that it would be a mixture of the properties that we now so confidently ascribe to either plants or animals. And the idea was not to say that there was no difference between plants and animals. There are obviously differences at the cellular and the macroscopic level. However, Bergson wished to remind us that as products of an evolutionary history, animals and plants are not static categories, but carry ever-moving dynamic properties. And we should therefore not be surprised when we come across an animal like coral that seems to blur the boundaries. And it's just a reminder of the fact that we ourselves are nothing but a phase in an evolutionary history. Around that time, a young zoologist at the very beginning of his career, by the name of Julian Huxley, started formulating his own philosophical ideas inspired by Bergson's metaphysical biology. And if you were wondering, he is related to Aldous Huxley, he's his half-brother, and he is related to Thomas Huxley, he's his grandson. So quite a lot going on in the Huxley family. Julian had been studying cycon, which is a calcareous sponge that, like certain species of coral, secrete calcium carbonate to form a protective exoskeleton. And he studied the regenerative powers of this organism. And one of his mentors, Henry Van Peters Wilson, had noted that for organisms like these sponges that they could see regenerate before their very eyes, the ordinary idea of the individual is not applicable. And this got Huxley thinking about what it meant to be an individual. What kind of an individual was an invertebrate, and how did it differ from being a human individual? In 1912, he published his first book, The Individual in the Animal Kingdom. And inspired by Bergson, he did his best not to consider the notion of individuality as a static category. Individuality needed to be understood as something that, like everything else in the living world, had evolved and was still evolving, and presented itself under different forms. It was not a fixed characteristic of animal life. There were degrees of individuality, and it had to be thought of in terms of process or tendency. And so with this fluid evolutionary conception of individuality, what counted as an individual transcended the habitual boundaries of the concept. For instance, the coral polyp itself could count as an individual, but so could the whole colony of coral polyps that formed the reef. In Huxley's words, there are colonies such as those of corals where a number of animals, each of which by itself would unhesitatingly be called an individual, are found to be organically connected, so that the living substance of one is continuous with that of all the rest. Sometimes these apparent individuals differ among themselves, and their energies are directed not to their own particular needs, but to the good of the colony as a whole. Which is the individual now? Why not even, said Huxley, think of human societies as individuals? Like the coral colonies, the different parts of a society, so each person, can be considered as individuals in their own right, but their common and collaborative action also strives, or should strive, towards the conservation of the whole. That's something to think about, and I think Ellen is going to say a lot more about the notion of individuality, but now that I've delved into the murky depths of mythological biology with Ovid, geological biology with Darwin, and philosophical biology with Bergson and Huxley, I'll pass on to Ellen, who will introduce some more current problems in the philosophy of biology.
Okay, yeah, so returning to the contemporary scene now, Huxley's question, which is the individual now, certainly hasn't gone away. There's a very lively debate in biology and in philosophy of biology now concerning how to decide in different lineages which lump of stuff counts as one individual, as one living thing, rather than many. So if you look around the living world, one thing that humans tend to notice is it's often subdivided into discrete packets. We don't see a sort of living soup in front of us, but separate, countable organisms. And the organisms that we interact with most often tend to come with a suite of features that make them really easy to distinguish from one another. So they have skin or bark wrapped around them, sort of marking their boundaries. They have immune systems which police those boundaries and sort of stop outside stuff from getting in. Their life cycles tend to have clear start points, and each organism tends to have its own unique genome. Then there are non-paradigm cases. Cases where, as Huxley's advisor wrote, the ordinary idea of the individual is not applicable. Now, marine invertebrates are an excellent source of these. They're all creatures where it's hard, for a number of reasons, to say where one ends and another begins. So sponges, as Emily already mentioned, can be cut up. They can actually be sieved and survive sieving, and after being sieved, their parts will coalesce and reform a functional individual. There's ascidians, like the organisms on the right there, which spread across the surfaces of rocks. And where one ascidian meets another one, at least some of the time, they just fuse. Their circulatory systems fuse, and they become, in some sense, parts of a single organism. Starfish. There are some species of starfish that reproduce by ripping their arms off and regenerating the missing parts around the arm. So corals are in really good company as non-paradigm organisms. And there's two particular problems that I'm going to talk you through. Coloniality, that Emily's already talked about a little bit, and symbiosis. So first, on to coloniality. As a matter of fact, some corals are solitary, but many of them are colonial. So once it's found a place on which to do its handstand, a coral polyp starts to bud, make a clonal copy of itself. So either the polyp rips itself in half and regenerates the two halves of the polyp, or a sort of outgrowth of cells forms on the side of the polyp and grows from there, which is what you can see diagrams on the right. There are actually nerve connections between neighbouring polyps. So if one polyp is disturbed in some way, you can see a wave of retracting tentacles passing across the colony as the signal is passed between its neighbours. Now, it's absolutely no surprise that earlier generations found it difficult to distinguish corals from plants if you look at their growth forms. Branching corals, in particular, have a very similar appearance to trees. And the analogy is actually really deep, because trees, in some sense, are colonial too. So in both cases, this branching tree shape occurs when small modules, so these are buds in the case of trees and polyps in the case of corals, grow in a sort of repeating cycle where each bud or polyp grows on top of its parent. And like the parts of trees, the polyps of a coral colony are all genetic clones of one another. So interestingly, Darwin's grandfather, Erasmus, he advocated treating trees as colonies of buds, where what happens each spring is you get a new generation of buds being born atop the branch that's been left by its parent. So there's a sort of Necker cube effect here, where we can choose between two available perspectives. The question is, should we treat polyps in a colonial coral as separate organisms, or should we instead treat them as mere parts of a single organism, like we do with something like an oak tree? Now, the choice, popularised by Daniel Jansen in 1977, has us view an organism as including anything that's cloned from it, so that the whole coral colony is just one organism. He argues that natural selection can't distinguish between things that share a common genotype, so neither should we. Even when the clones are spatially scattered, as with dandelions, even when the clones are completely unattached to each other, as with aphids, Jansen claims that they're all parts of a single organism, really just as much as my arm is a part of me. A genetic view of corals, then, would treat a whole colony as a single, very long-lived organism, rather than as an extended lineage of many generations. And it's important here that these two perspectives yield different implications for some important questions, such as how old corals are and the rate at which they evolve. Very few researchers today would follow Huxley in thinking of whole human societies as organisms. But there is a sense in which humans themselves are colonies, of course, colonies of billions of cells. Multicellularity is a phenomenon that evolved out of coloniality. So really, coloniality is not a niche phenomenon here. Coral colonies just serve as a particularly beautiful illustration of a trait that's actually very common. This is not to say, however, that humans and coral colonies have the same status. The parts of a coral colony are much less differentiated from one another than are parts of a human, for example. So most of my parts have no autonomy at all. They can only survive and reproduce as parts of me. They can't survive and reproduce on their own. So we can distinguish between aggregates that have become true organisms from aggregates such as societies and coral colonies, where there's much less connectivity and interdependence of parts. So actually, I don't think we should treat whole coral colonies as organisms. I think the reductive view that takes the polyps as organisms is right. But the lesson that coral have to teach us here is really that there aren't sharp lines between colonies and organisms, between parts and wholes. Nature instead offers us a whole spectrum of forms, a gradient between one and many. So, on to the second of the contemporary puzzles about individuality. It's a fact, apparently, that cold water supports more life than warmer water because colder water contains more dissolved oxygen. So warm water tends to be clearer because of its sterility. There isn't any sort of drifting plankton there to cloud the water. When I heard this fact, it struck me as strange. I wondered why, when I think about tropical seas, I always imagine this sort of colourful underwater paradise of life. Well, the answer is coral symbiosis. Corals essentially figured out a way to flourish in what should be an uncomfortably warm, sterile environment by getting into a partnership with a kind of microscopic plant. So, known as symbiodinium, or more commonly zooxanthellae, these are tiny algal cells that live inside the tissues of the coral. And they actually photosynthesize to provide the coral with most of its energy as well as oxygen. And in return, the guest workers get, well, comfortable housing, they get protected inside the tissues of the coral, and they also get a supply of carbon dioxide and other nutrients that they need. So the lesson is, if you ever find yourself needing to survive in a glass box with no food or air, get yourself some plants, and you might be all right. Corals aren't the only animals that have taken on domestic help in this way, so one of the most striking examples is this sea slug, or Elysia chloratica, that's truly part animal and part plant. The green colour that you can see here in this sea slug is actually from chlorophyll, which is produced by chloroplasts. Now, the chloroplasts are found in the algae, which the slug eats, but the slug has a really simple stomach, so when it eats the algae, instead of properly digesting it, it's actually taking the chloroplasts into the tissues of its own cells, where they carry on living and photosynthesizing. And the stolen chloroplasts can actually be so efficient that these slugs can live as long as 9 months without eating anything and carry on growing and being quite happy. So, as well as their plant pals, corals house all sorts of other microbial symbionts. They've got bacteria in the mucus that covers their polyps that gives them antimicrobial protection. They've got all sorts of other microbial symbionts, and they also provide food and habitat for a huge number of other species, including fish. So it's really thanks to the energy exchanged in that partnership that coral reefs are like underwater rainforests. This would otherwise be a sort of sterile water column, but they fill it with these complex three-dimensional structures which provide homes for all sorts of life forms. According to one figure I read, about a quarter of all marine biodiversity occurs in coral reefs, something like 4,000 different fish species. Now, although, as Emily told us, people were interested in individuality at the birth of the modern synthesis, that synthesis almost completely ignored symbiosis. There have been lots of calls in recent biology and philosophy of biology to change this, to shift to a more cooperative view of life, one that looks beyond the sort of competitive struggle for existence, to really acknowledge just how much living things depend upon one another. Some researchers are advocating use of what they're calling a holobiont concept. So a holobiont is a host plus all of its symbiont microbes. And the hologenome is an associated concept. It's just the genes contained in the host plus the genes of all its symbiotes, considered as a sort of shared resource for all those parts. And defenders of this view claim that instead of treating coral polyps as single organisms with hundreds of cellular symbiont organisms, we should treat them all as part of a larger holobiont organism. So, corals, according to this view, are aggregates, coalitions of disparate species that have been treading a common path a common evolutionary path for millions of years. So in this sense, they're neither plants nor animals, but they're truly both. And once again, this sort of conceptual acrobatics might seem easy to dismiss as somewhat niche, as applying only to fairly primitive or marginal life forms. But again, corals are here illustrating a phenomenon that's absolutely central, because human beings, the most paradigm of all organisms, are coalitions also. So, way back deep in our evolutionary history, before we were multicellular, an ancestor of ours took on domestic help, just as the corals have done. So our ancestor cell was a kind of bacteria, and it engulfed another cell. And instead of digesting it, it became a permanent symbiosis, what we call an endosymbiosis. The image that you can see on the right there is a diagram of a human cell. And the things that sort of look like yellow slug shapes are called mitochondria. So these are things that produce all of our cells' energy. And each of us has got hundreds of these little evolutionary hitchhikers inside each of our cells. And actually, we're even more exotic than plant-animal hybrids because they think that our mitochondria came from a whole other domain of life, the archaea. So the modern view here is that there are three domains of life, but the eukarya, where plants and animals are found, were actually formed by a merger between the other two domains when all of the endosymbionts, like mitochondria, were taken on. So we're all hybrids. In addition to this, humans carry so many bacterial symbionts in our guts and on our skin and in other places that there are thought to be as many bacterial cells in a human as there are cells with human DNA. So, instead of thinking of humans as colonies of genetically identical cells, some philosophers are now endorsing a radical view in which a human is viewed as a holobiont. So it's composed of millions of gut microbes, viruses, all sorts of other pathogenic and sympathetic symbionts, in addition to the mitochondria and the cells that contain human DNA. And parity of reasoning there might also seem to imply that we should treat corals' algal partners as true parts of the coral, just as we classify mitochondria as parts of our cells. And again, I don't think we should be so radical. There are important differences between relationships that are permanent because the interactors are sort of joined together throughout their lives, throughout their births and deaths, as is the case with mitochondria. And interactions, on the other hand, that are more fleeting or contingent. So corals don't actually keep the same zooxanthellae all their lives. The algal partners can come and go during the lifetime of the coral polyp. And this has got some really important consequences for the way that the pair evolve. But again, it's a crucial point that these differences are differences of degree. They're not differences of kind. There will always be borderline cases, in-between cases, messy, ambiguous, confounding cases. And all of these are the cases, I think, that make life so beautiful. So, corals are gorgeous, but they also have lessons to teach us. They teach us that individuality is not something fixed to be presupposed. Organisms didn't get put on the planet fully formed. Instead, they evolved gradually. So borderline cases are inevitable. Corals remind us that the history of life is a history of mergers, of aggregations and intersections between different branches of the tree, or more properly, the web of life. And finally, corals remind us that there are no neat lines in biology. So whatever boxes we invent will never persuade life to fit neatly inside them. So it's time for the sad ending for the talk now, because just as we're starting to appreciate all of the amazing lessons that coral have to teach us, they're disappearing. So corals are under pressure from a number of threats. They're suffering from invasive predators such as crown-of-thorns starfish, from pollution, from sedimentation that occurs after coastal deforestation. Destructive fishing methods are damaging them, things like trawler fishing, and coastal developments where people reclaim land are destroying their habitats as well. Most of all, climate change and its associated rise in sea temperatures and acidification of the oceans are a problem for them. Like the British, in hard times, in times of stress, corals seem to make the bizarre decision to eject all of their guest workers. And as with Brexit, it's probably not a good idea. So the corals eject their algal symbionts when they're stressed by things like high temperatures. And without these symbionts colouring their tissues, the polyps are transparent, so you can see their white skeletons through. So the corals look bleached. Without their symbionts, the polyps are weakened, they're vulnerable to disease, they grow only slowly. Sometimes they regain their symbionts when conditions get better, but if not, they die. Australia's Great Barrier Reef, where about 10% of all reefs are found, thought to be about 20 million years old. It experienced what's called a severe bleaching episode in 2016. Most of its coral bleached, and 22% of it, something like that, subsequently died. So bleaching events have been known before. They happen sporadically. Usually the corals get years to recover after the oceans cool again, and they regain their symbionts. This year's seen an unprecedented second severe bleaching episode in 2 years. So we don't yet know how many fatalities from this year's bleaching episode. Corals are often taken as barometers of oceanic health and global warming more generally, and the signs are not good. By 2050, experts are predicting that probably most of the world's coral reefs will have been destroyed. So as not to have too depressing an ending, there are some ideas out there about how we might save the corals. So for example, marine parks are being created in which corals are protected from things like destructive fishing and from pollution, although it can't really save them from climate change. Coral nurseries, like this one in the Seychelles, are being used to try and restore reefs. So what they're doing here is capitalising on those regenerative capacities of corals by taking fragments from healthy corals and then growing them in protected conditions before transplanting them back, either to boost polyp numbers in an area or to found new reefs. The idea behind designer reefs is to capitalise on existing variation in coral resilience to stress of various sorts. So corals are very different, and so are the symbionts that they live with. So there are corals in the Arabian Gulf that can withstand temperatures of 35 degrees. They don't mind it hot at all. There are other corals that grow in really cold water, in really cold deep water, and don't bother with algal symbionts at all. So there's a lot of diversity in the population. What designer reefs would be doing to exploit that diversity is basically transplant corals into locations that are better suited to them or to their algal partners. And finally, coral probiotics. So just as advertisers have for ages now been trying to persuade us that we need to drink these yogurt drinks to get good bacteria to protect our digestive systems, which is nonsense and doesn't help at all. But actually, scientists are advocating the use of probiotics more sensibly to help cope with thermal stress. So coral reefs would basically be inoculated with selected zooxanthellae and other beneficial microbes, particularly strains that are known to cope really well with hotter conditions. So you give them some new partners, essentially. So corals have literally redrawn the map of the world. They've built islands big enough to need airports. And they've done this by building on top of the skeletons of their own dead ancestors, as Emily mentioned. So they're tough. And we can see from the fossil record that they've been through bad times before. So if you can make out the green lines on this figure, they show where there have been corals in the fossil record. So corals first appeared in the late Cambrian, something like 500 million years ago. They subsequently disappeared from the fossil record something like 9 times. So there are blank spaces here, hundreds of millions of years long, where there are no fossil corals at all, only for them to reappear later on. So each extinction event that you can see here is associated with some sort of global shock. So there's a thermal event, or a meteor strike, or an ice age. So corals have been through hard times before, and although they disappeared from the fossil record, the lineages weren't lost entirely, and they later made a comeback. Evidence suggests that corals have now gone into another terminal decline and are going to disappear. On the other hand, they're damn adaptable little critters, and it's really precisely those features that make them so resistant to our sort of conceptual tidying up activities. So their amazing regenerative power and their cunning use of these symbiont partnerships, it's those features that have enabled them to make so many comebacks. So if I want to end on a positive note, we can note that corals probably stand a better chance of recovering from the destruction that we're wreaking on our planet than we do in the very long run. So thank you for listening. And so that you don't go away feeling too sad about the end of humanity, you can start getting excited about the next instalment of HBS in 20 Objects, which is going to be on the 23rd of May, which will reveal all sorts of wonders about a Bar and Stroud rangefinder. So we can look forward to that. Thank you.